Thank you very much for the opportunity of giving to me to talk about what we are doing. And this is not too new to you, actually, the chair. I talked about this couple of years back in Chicago. How do I get it started? So what I'm going to talk about is what we refer to as photodynamic vaccination, and using non-viable but vaccine delivery competent leishmania, and which is inactivated by loading with photosensitizer and then excited by light, which will generate a free radical, especially the synchronous oxygen. And so the organism is inactivated. And as you know, leishmania belongs to trypanosomatid protozoa. It's a parasite. It does have a number of advantages to serve as a vaccine carrier. And so I'm going to talk about this a little bit before I get into the main point. And as most of you probably know, leishmania, certain species can produce innocuous, self-healing, self-limiting skin infection. And that amounts to effective immunization, eliciting a lifelong immunity to reinfection. So at least some species behave that way. And this could provide a favorable environment if you put in some additional vaccine and can elicit better immune response against the new vaccine you put in there. So the other point is leishmania can be cultured in the lab in fair quantity with ease. So what you see here is the so-called promasical stage, as some of you know. These are in the insects. These are transmitted by the insects, and this can be grown. So as you also know, these are eukaryotic cells. So they have a nucleus, and they have anterior flagellum. So this stage is extracellular. And then you can culture this, probably very important in terms of manufacturing in chemically defined medium. So without any animal body fluid like fetal bovine serum, and they can be grown in quantity in fermenter, is the published information. So this is the starting material, can be used to deliver vaccine. So the third point is leishmania in their natural infection. They have some homing specificity to the phagolysosome of antigen presenting cell. So their normal host cells are the macrophages. In addition, they can infect dendritic cells in which they don't really grow, but they can infect. So this is supposed to be a desirable destination for the vaccine processing and presentation. So this is just a picture to show you the general idea. You culture these promasicals. This is the electron scanning EM picture. And then you can use this to infect macrophages in culture, macrophage cell line, peritoneal macrophage, monocyte from peripheral blood, or in vivo, injecting into skin. And so what's happening is a large number will bind to the surface of the host cells. For some species, they can get into this phagolysosome in the form of a very large vacuole. So we use these species for the reason it's easier to assess. And so they will differentiate into emasicals. So if you transgenically put in the GFP gene, so you can make them green fluorescent, and you can do the quantitation easily. So naturally, Leishmania can do this very effectively, and they can make use of the host molecule in the body fluid and to the advantage of getting into the macrophage. In contrast to many parasites or pathogens, they don't want to be there. And this is so happen to be the destination of their site for growth. So here what you see is the promacigos. In the insect gut, extracellular, when they get into the macrophage, they become smaller, imacigos. So this is the condition of changes. And so in the process, they have some surface glycoconjugate, carbohydrate, lipid, protein complexes, and this is very important molecule for the parasite resistant to the microbicidal factor in the body fluid, and in addition, they are responsible in a large part for the homing of this organism to the phagolysosome of macrophages. And so these are the conjugate. They may well serve as potential vaccine adjuvants, because in the natural infection, you get a cure and immunity for life. There's no additional adjuvant needed. So this comes to the main point. How do you inactivate this organism so they can be non-pathogenic, and yet they may still be very useful to use their natural ability for targeting to the APC. And the way we're doing this is by oxidative photo inactivation. And so I'm going to explain this a little bit. And we are doing this in two different ways, involving the use of photosensitizer. So these are ring compound, light sensitive, and once you eliminate with light, they generate single oxygen and so on and so forth. So we're doing this in two ways. One is chemical engineering by loading the promacigal stage. You can culture with the photosensitizer, and then the other is genetic approach. By using their inherent deficiency in heme biosynthesis. As you know, in the heme pathway, there are porphyrin as intermediates, which are photosensitizer. So I'm going to talk about this a little bit more. As the picture you see here are three examples of photosensitizer, hyperrhizine, porphyrin, and phthalocyanin. So these are representative in the sense hyperrhizine is coming from plant. So there are many plants that have this dye or pigment. The porphyrin is intermediate in the heme pathway, and the phthalocyanin are synthetic compound. You can prepare this in the lab synthetically. So their interaction with the Leishmania promacigos is shown here. This is the phase contrast, and they can load the membrane. So once you put this in, you can see the fluorescence of the photosensitizer immediately in the membrane, and this is also true for aluminum phthalocyanin, and the porphyrin needs to be induced by genetic transgenic Leishmania. So the point here is not all known photosensitizer can be used to load cells. If they are not loaded, then they are not light sensitive, so they will not be effective. So it is the main point of this one. So we're working with Graca Vicente. She is a chemist, a professor in Louisiana State University. So she's making the phthalocyanin compound with different metal and ligand, and also side chain. And so she provided us with a series of this material, different kind of ligand, different metal, zinc or silica, and the different length or the bulkiness of the ligand, and this will change the property for interaction with cells, not only with Leishmania, but also with mammalian cells. So the picture here is showing one example. If you increase the ligand in this pyridyl thyrocyanin, increasing length of the ligand and increasing pegylation, then you can see they are increasingly capable of being taken up by Leishmania. And once you have more accumulation, they will be more light sensitive and more free radical can be generated. Then you can easily inactivate them. So here is another picture showing a different thyrocyanin, showing their uptake into the endosome in both the extracellular, intracellular stages of Leishmania. And so once you load them up, then you can illuminate them with light. The more photosensitizer, they are more light sensitive. You can use very dim light, and to the point of luminescence is enough, as long as you have the right wavelength. So what we are using here is GLP transfectants. So this is the control. You can see they are intact. They are fluorescent. There's no thyrocyanin fluorescence. And you can load this with the PC 1 and 2, so you can see they're loaded. If you don't illuminate with light, there's no changes. If you illuminate with light for as short as 10 minutes, you can see they become disintegrated, inactivated, but they are not falling apart. So this remains as a particle, and you can use this to deliver drugs or vaccine. So the other approach you are using is a genetic approach, and to accumulate photosensitizer in this group of organisms. So this is based on the genetic complementation of Leishmania inherent deficiency in the heme biosynthetic pathway. These are aerobic organisms, so they require heme, much like us or any other aerobic organism, but they are not able to produce this, so they need to uptake heme in some form, hemine or something else, in order to grow. And so when the transgenic process become available for Leishmania, we did the transgenic approach to produce the enzyme among these 8, which is the classic pathway enzyme for heme biosynthesis. Many years ago, we thought they might have some of this enzyme, but when the genetic project, genome project is completed, we know they don't have probably everything except the last one, ferrochelatase, for some reason still stay there. So we are putting in the gene, cDNA from mammalian cell, coding for the second and third enzyme in 2 different plasmids. So both of these are shuttle vector between E. coli and Leishmania. The thin one represents nothing but P blue script. So you can expand this in E. coli using the ampicillin resistance. The thicker area are the DNA from Leishmania. They require certain sequence, and you need a different selectable marker. And then here are the gene we can insert. And then we can do electroporation, we can select. We can do this at the same time. So once you have the gene put in, essentially you have enzyme 2 and 3 expressed. But there's no first enzyme, so if we supply with the product of first enzyme, the ALA, this is delta amino levulinase, and then they can be taken up easily. Then with these 2 enzymes transgenically introduced, they will produce the porphyrin. And then you can have a inducible accumulation of a photosensitizer in the form of uroporphyrin in these pramazigos, and this becomes extremely light sensitive. If you have uroporphyrin incubation of the parasite, they are not taken up at all. These are very water soluble, and so you need to do this and making them very light sensitive. So if you have this transgenic leishmania, you can treat them with ALA, feed them to the macrophages. If you don't eliminate, they remain intact in the vacuole. If you have the light, then you can see they become disintegrated. So here is another control showing they are being intact. So, so these are the two different ways. We are photosensitizing leishmania and inactivate them by light. And why you want to do this? Because the single oxygen is the major product of this photosensitizer, porphyrin, thiocyanin. They are very short-lived. So they are in the, in the cell. They can rapidly inactivate all the vital enzyme, but they cannot cross membrane. So the surface conjugates remains effective, serving the function of homing and protection of whatever the vaccine you can put into leishmania. So the next point is about the safety of this material, and we did some experiment to prove this. So if we do the double photosensitization, loading them externally with thiocyanin, inducing accumulation porphyrin internally, then you can show there's nothing come up to 8 weeks or beyond. And the others are all controlled. And you don't produce any skin infection when you have this doubly photo-inactive leishmania as a potential safe delivery of a vehicle for vaccine or drugs. So the first thing we want to try is, of course, using a typical antigen like OVA. And yesterday, the number of immunologists talking about using this. So we also approach this, and we've done a fair amount of work, but I'm not going to present all of them. So the in vitro model, you could have OVA delivery by this oxidatively photo-inactive leishmania to dendritic cells, APC, and then you can show the activation of the CD4, CD8 specific T cells. And so here is just one example. We are doing this in culture. So for this kind of work, there are a lot of control. I'm not going to go over all of this except the first three. So Symphicol is the peptide, and this is OVA. You can see they can activate the T cell line. And the third one is the OVA delivered by leishmania photo-inactivated, at least in the bone marrow macrophage. They appear to do very well, and even in the dendritic cells. These are cell line. They are better than the other control. And we have some animal data. I'm not going to show this. So basically, antigen like OVA can be delivered in this way, and after photodynamically oxidized, and they appear to retain not only their immunogenicity, but also it appears 5 minutes. So then I have to move fast. So we did some vaccination against the visceral leishmaniasis in hamster. So these are the parasite burden decrease. And after vaccination, you see the interferon gamma. All the good cytokine appear elevated, and bad one decrease. And this is can be adaptively transferred from the lymph node cell into naive animal, basically showing the same thing. and you have this good cytokine elevated, and the others going down. And we've done a number of different lab animal study, and a lot of these not yet published, and we are making a new generation of photo-sensitized leishmania. So the other thing we're doing is in treating canine leishmaniasis in Naples, and it is an incurable disease by drug, and 50% of the drug-treated dog will eventually develop canine leishmaniasis again. So we are trying to do immunotherapy after chemotherapy, see if this can cure them. So this is just a map showing some work we are doing in Brazil with a breast cancer surgeon working on cancer, vaccine delivery, Italy, and other places. So here is some picture to show you the canine leishmaniasis data. And finally, we have some cancer vaccine candidate expressed quite well in leishmania, and we want to try this in collaboration with others. So here is the PRAM expressed by leishmania in the Western blot. And so to summarize, so this is a new way to deliver vaccine with efficacy based on the unique property to homing to the APC and so on and so forth. And they're safe if we do this doubly with two different ways. So this is what we would like to collaborate with the colleagues to develop this, to try this out. Okay, so I finish in 5 minutes.